Hello, I'm Dr. Charles Knight, and this is my series of lectures called 10 Lectures on Evolution that I'm recording for my evolution class, Bio 414, and uh, I work at Cal Poly in San Luis Obispo, California, USA, and I work in the Department of Biological Sciences. So I've got some other lectures in this series I hope you can link to, and feel free to share and enjoy. This is our target paper number 3 that I have chosen for our class. It was published on October 21st of 2024, and it's titled Tolerance and Efficient Metabolism of Extremely High Ethanol Concentrations by a Social Wasp. It was published by these people, Sophia Bucciabetti and Eran Levin, at Tel Aviv University. Right now, Sophia works at Ben Gurion University, and if you there's a nice biography of Sophia on the internet that I found that's linked here in the PowerPoint. On YouTube, you won't be able to see it, but it's pretty clear to me that the oriental wasp is a fascinating insect for the investigators. I have a feeling that it must colonize their balconies, maybe even right there at their university or their houses there in Israel. And these wasps enjoy alcohol, or they tend to congregate around alcohol and they consume it, and that's really what this paper is about. I want to mention that I'm going to present about the target paper, but there was a commentary paper in PNAS in the same issue that really helped me in this presentation. And it was authored by Rainier Spanagel from Germany, from Heidelberg University. He works at an institute there for psychopharmacology. And again, there's resources there, and he's got a really interesting research program. And this commentary really kind of helped me outline this presentation. So I'm going to start with Rainier's story, and then I'm going to go into Sophia and Iran's paper in quite a bit of detail, and then I'm going to go back and finish with Rainier's story here on how to survive enormous amounts of alcohol. Because Rainier's story has more to do with the human dimension, and I think a lot of you and a lot of my students might be interested in that. In addition, I'll have some textbook tie-ins along the way, and the textbook that we use is Evolutionary Analysis, the 5th edition by John Heron and Scott Freeman. And of course, I use Wikipedia and Google as well to get more information. Just to keep you interested, there'll also be some vignettes on my own tattoo and my own AUD. So we'll touch base on that at the end of this presentation, or in the middle too. So I always have take-home points at the beginning of these presentations. Just want students to know what's important, or just some things to take away from these papers. The first is that highly efficient ethanol metabolism in oriental hornets is most likely due to a duplication of the alcohol dehydrogenase gene, or ADH. We're going to see that there's some hornets that have 4 copies, but the oriental hornet that they studied likely has 2. We'll take another look at that in a little bit. Wasps and hornets may benefit from the 2 times higher energy content of ethanol compared to glucose. So there's more energy in this ethanol, as well as from its antimicrobial properties when feeding on carrion, on dead animals. Maybe there's some sterilization going on there with feeding on this alcohol. And of course, the yeasts are benefiting through this mutualism by dispersal to new sources of glucose, new rotting fruit. The wasps and hornets are seeking out this rotting fruit to get this high energy compound, and in the process are dispersing the yeast, so there's further fermentation happening. And wasps actually use the energy from ethanol, and this is one of the real main take-home points from this paper. They used a stable isotope analysis to show stable isotope of carbon that they enriched an ethanol source with actually showed up in the CO2 that was breathed out by these wasps, and they tested it on bees too and didn't see that. So that was a big result from this paper. Fourth is that AUD in humans may be partly explained by slow ADH enzymes, partly. AUD is alcohol use disorder, or another way of saying, or a new way of saying alcoholism, which carries a lot of baggage. So Ranier's paper deals more with AUD in humans and this slow ADH enzyme. And lastly, while ADH copy number and allelic variation may explain hornet ETOH tolerance or ethanol tolerance, humans have more copies of ADH and cannot tolerate 80% ethanol like the hornets can. And so there must be other factors at play. And for my students, I think this is a really important point that this paper, the primary literature paper that I'm discussing by Sophia and Iran, is a starting point, but it's a really good starting point for understanding ethanol metabolism in insects and ultimately in humans too. So there's more to the story, and it may involve other enzymes too, but ADH plays a role for sure, and copy number variation is an interesting aspect of that. I always read the abstract first. Authors put a lot of effort into this abstract, so we should start there. Ethanol, a natural byproduct of sugar fermentation, can be found in various fruits and nectar. Although many animals routinely consume ethanol at low concentrations as part of their natural diets, its inherent toxicity can cause severe damage. Even species particularly well adapted to ethanol consumption face detrimental effects when exposed to concentrations above 4%. Here we investigated the metabolism of ethanol and its impact on survival and behavior in the oriental hornet, Vespa orientalis, a social wasp that naturally consumes ethanol. We show that chronic ethanol consumption, even at concentrations as high as 80%, had no impact on hornet mortality, construction behavior, or agonistic behavior. Using C13 carbon 1 labeled ethanol, we show that hornets efficiently metabolized ingested ethanol and at a much higher rate than honeybees did. The presence of multiple copies of the alcohol dehydrogenase NADP+ gene in the Vespa genera suggests the potential mechanism for the ethanol tolerance. These findings support the hypothesis that the mutualistic relationship between ethanol-producing organisms and vespid hosts may be at the origin of the remarkable capacity to utilize and metabolize ethanol. As I do with an abstract like this for my students and even just the general audience here, it's important to familiarize yourself, I think, with some words, and those words I have highlighted here, or I circled. Vespa orientalis, 80% ethanol, and this C13 carbon, alcohol dehydrogenase, NADP+, and mutualistic relationship are all key terms that I'll define on the next slide. I use a presentation like this where I actually define those terms in what I call glossary notes. So Vespa orientalis, again, is a social wasp that naturally consumes ethanol that's native to the Middle East and Israel. So it was around Sophia and Iran when they were thinking about this paper. And I think that that's how they led to it. 80% is the ethanol concentration Vespa orientalis can tolerate without negative effects. They showed that on construction behavior. They kept on building. They didn't have more agonistic behavior, and they survived actually better with 80% ethanol. So they were actually using this. 4% is the ethanol concentration most other organisms can tolerate without negative effects. So you can't tolerate that 80% ethanol, and how they can do that, how these wasps can do that is the question. C13 carbon 1 labeled ethanol. This is ethanol labeled with C13, which is a stable isotope of carbon with one extra neutron that they could detect in exhaled CO2. So they had a mass spec or some kind of instrument that could easily detect this. I said easily and I deleted that word. I'm sure it's not easy, but they could detect it, which is pretty cool. What I always say to my students is that once you learn how to do stable isotopes or you get into that, you don't come back, because you can discover so many cool things in ecology and evolution and physiology using stable isotopes. And here's just a diagram of ethanol on the left-hand side, with one of the carbons having C13, which is that carbon with an extra neutron, And then once it's metabolized, it converts, of course, it produces this carbon dioxide that then also has this C13 that they can detect. And it's really that molecule on the right, the CO2 with the C13, that they're detecting and showing that the wasps actually were using this ethanol. Mutualistic relationship is just when both species benefit, both the wasp and the yeast. And then lastly, I just want to highlight that ADH-NADP plus is a type of alcohol dehydrogenase that reduces NADP plus as part of its alcohol reaction. Whoops, I misspelled alcohol there. There are other types. There's an NAD plus ADH as well as an NADP plus ADH. So they were studying this ADH-NADP plus enzyme. So when I go back to Ranier's paper, How to Survive Enormous Amounts of Alcohol, there's some take-home points that I just wanted to help you, or to begin the story with. And Ranier's paper was really very helpful for this. Alcohol dehydrogenase catalyzes the conversion of ethanol to acetaldehyde. This is basic biochemistry. This is ethanol. Again, second time I'm showing you a carbon structure of ethanol. 2 carbons in an OH group, basic organic chemistry. That's your ethanol. Here's alcohol dehydrogenase, a picture of the enzymatic structure. Not that important for our study, but we know a lot about alcohol dehydrogenase. It's one of the most studied enzymes out there. Besides ribisco, alcohol dehydrogenase is one of the most studied, I would think. And that enzyme, using the substrate ethanol, produces acetaldehyde. And acetaldehyde chemical structure showed there. You have an aldehyde group, the C double bonded to an oxygen, and a hydrogen. That's the aldehyde group at the end. And as we'll see, this acetaldehyde has consequences for the metabolism and physiology of organisms. And it makes people not feel well. And that's why a lot of people don't drink as much, is that they accumulate acetaldehyde perhaps faster than other people. And that is a natural prophylactic against drinking too much, of course. There's more to that story. So acetaldehyde dehydrogenase converts the acetaldehyde to acetyl-CoA with coenzyme A. And once it gets down to coenzyme A, or sorry, acetyl-CoA, that's good, because this is the acetaldehyde dehydrogenase enzyme, and acetyl-CoA, that's good. That's really a starting point for lots of metabolic reactions, the acetyl-CoA. So if the ethanol gets all the way to acetyl-CoA, that's generally a good thing, because that's a productive entry point into the rest of our metabolism. In most organisms, genetic variations of ethanol metabolizing enzymes such as ADH modulate the effects of alcohol and thereby influence the amount of consumed alcohol. So in humans, there are 7 different ADH genes with several different isoforms that have been identified, and these are referred to as ADH1 through ADH7, but it gets a little complicated, the terminology here. But actually, before I get into that, the isoforms of these enzymes, the ADH1 through 7, have different Michaelis-Menten kinetics. And so I'm just reminding you that Michaelis-Menten kinetics is one way of describing how fast an enzyme is working based on a substrate concentration. And Km values for ethanol vary by a factor of up to 5,000 for different ADH enzymes and for different organisms. I'll just remind you again in the glossary notes here, the Michaelis-Menten kinetics it's a differential equation describing the reaction rate V, which is the rate of formation of a product P from a substrate lowercase p, to A, the concentration of the substrate A. So you can see it in a graph where substrate concentration A is on the x-axis and the rate of reaction V is on the y-axis. And Michaelis and Menten were like, these curves are all varying. You test this one enzyme, you test this other enzyme, and they have a different curve, and you test this other, things are just so variable. How are you going to compare these curves? We need a quantitative metric. So they thought, well, what if we do 50% of the maximal rate? And the concentration, the substrate of the concentration A, the substrate concentration of A that produces that 50% of maximum rate. So that's Km. So Km varies by an order of 5,000 in for alcohol dehydrogenase enzymes. So that's quite a bit of variation. So greater ETOH or ethanol metabolism can be achieved by better enzymes or having more copies. Better meaning just different Km, or just having more copies of the same ADH enzyme. And it turns out that hornets have duplicates. If we look across the animal kingdom, we can see the humans up there on the right-hand side have ADH1 through 7. ADH1, it gets confusing because ADH1A, 1B, 1C, and that'll become important later. I wouldn't have pointed it out if it doesn't. But humans, the way that they've labeled these ADH enzymes in humans are, as you see it up there. In hornets, they have two copies, only 2 copies, and compared to humans with their 7, but they have twice as much as the honeybee, which is right below the hornet on the left-hand side. You can see other organisms, they vary in copy number for ADH. C. elegans above the hornets there also has just 1 copy, but the hornets have 2 copies, and that might partly explain why they can metabolize ethanol so well. So I'll get back to the target paper here now. I was just lecturing a bit about the intro information presented by Rainier in his commentary paper in PNAS, in the same issue here as our target paper on tolerance and efficient metabolism of extremely high ethanol concentrations by a social wasp. And I'll just say it again, that the highly efficient ethanol metabolism in hornets is likely partly due to the expansion of the alcohol dehydrogenase gene family in wasps and hornets. I should also say that wasps and hornets are different organisms. They're on the same kind of evolutionary branch, but they're not within the same group. Well, I mean, they're different. I often use those words interchangeably here in this presentation, but realize they are actually different insects. The hornets are larger, and they're ones that you don't want to get stung by, and it turns out they're better at metabolizing ethanol, or alcohol in general. Some more glossary notes. How do you get more copies of ADH? This is a critical question that we should all be contemplating. And unequal crossing over is an easy route into gene duplication. I say an easy route because there are other ways to achieve gene duplication, but unequal crossing over is a common one. And I'll just define it for you here, starting with recombination. The general common recombination is when, during prophase 1 of meiosis 1, when chromosome arms overlap to form a chiasma. Those words you don't want to have to remember. Chiasma is a crossing over point, and that happens about once per chromosome arm, approximately, at least in humans. But sometimes the alignment is off, and one chromosome gets a larger fragment from the crossover event than the other chromosome does. And that's unequal. That's where the unequal word comes into this, unequal crossing over. This is a common diagram that's used to explain unequal crossing over, and students still have trouble conceptualizing it. And I haven't found a good animated video of unequal crossing over. And for those of you who are out there, and if you could let me know by email or somehow, if you've got a link to a good one, I'd really appreciate that. But from my Google searching, I still haven't found a really good one that goes beyond just trying to explain this figure. But if you spend some time with this figure, you can see that at prophase 1 of meiosis 1, that's what's described here on the left-hand side, chromosome tetrads are aligning, and there's a crossover event on one of the chromosome arms. There's probably a crossover event on the other chromosome arm too, but we're just isolating it to that one longer arm. They align imprecisely, partly because there's probably, well, who knows exactly why in this case, because there's not tandem repeats yet. But once there are tandem repeats in chromosome 3, that can lead to even more unequal crossing over because you have more islands of synteny between just imagine chromosome 3 pairing up with another tetrad of a similar chromosome with multiple copies of C. That can lead to its own slippage and greater unequal crossing over down the line. So the just trying to turn on the light again. This is the mechanism for unequal crossing over, and one way that gene duplication may happen in organisms. All right, we're going to get into the figures now, and part of this class is in science communication, and I'm encouraging students that when they're faced with a figure like this, it's not that good for presentation purposes. It's a 4-panel graph, and the pieces are pretty small, and you don't want your audience to have to read this small text, of course. So we're going to have to zoom in on it, and I kind of try to highlight for my students how to do that. First of all, focus on one panel, and second of all, maybe introduce one piece of information, like we're looking at 7 different groups and the 7 different colors. Those groups are different ethanol concentrations, and there's 50 hornets or wasps in each group. So there's replication there. But still, it's very small. So I'm going to instead focus just on this part, make that part bigger, and with some creative cutting and pasting, screen grabbing, and cropping, you can actually make a discontinuous y-axis and focus in on the data a little bit better, and label the axes again with screen grabs, but just make them very large in comparison. So now we can see that this is time in days going out to 6 days, and the survival probability on the y-axis there, and anything less than 1 is a decline in the survival probability of those 50 individuals in the group. Even across 2 or 3 days, you'd expect some natural mortality, because these hornets only live about, well, a few weeks. It looks like about 60 days or so. So 6 days is actually considered a chronic alcohol consumption. In this study, they just fed these hornets. That was basically their only food source. And we see that there's variation across 0 to 1 to 10 to 20 to 40 to 60 to 80% ethanol. But 80% ethanol wasn't off the charts worse for these hornets. And actually, statistically, it wasn't that significant. The difference between these groups wasn't that significant in their larger model. And so the conclusion from this graph is that chronic ethanol consumption at concentrations as high as 80% had little impact on survival probability across a 1 week treatment. That's cool. Next graph, we're going to look at lifespan, changes in lifespan. And we see this is going out to 120 days. I'm highlighting 120 there on the bottom on the x-axis because it's a much, well, fairly longer time span here. And we're only looking at the 0% and 80% ethanol treatments. And you see the red line, that's the 80%. The hornets actually live longer with 80% ethanol. My assumption is that there's more energy in that ethanol, and so they actually are getting a better food source. The 0% ethanol hornets are dying around 60 days. That is, the survival probability plummets to almost 0 after 60 days, whereas the hornets that are consuming ethanol are living much longer, almost twice as long, perhaps. Not on a population level, there's still quite a bit of mortality across 60 to 90 days, but some of the flies actually lived out to 120 days. So the conclusion from this graph is that chronic ethanol consumption also had little impact on hornet lifespan. These are interesting findings that show that ethanol doesn't have much effect on lifespan or survival along short or long timescales. This figure is a little bit more complicated. There's 2 groups of bugs here, bugs or flies or hornets and bees actually. On the top in the yellow and brown, I'm looking at the legend here first. Stare at the legend with me first before we look at the graph. The honeybees in yellow and brown. The hornet is in purple and red. The brown and the red are 80% ethanol. The yellow and the purple are 0%. What we see here, if we look at the brown, the 80% honeybee graph, those honeybees die. They die within 1 day on 80% ethanol. Whereas the 80% ethanol for the hornet, they survive much better compared to the honeybee. And for the 0%, both the honeybee and the oriental hornet survive about the same amount. And I think this was when they actually tethered the bugs and force fed them. The mortality is much earlier for this graph, but the point here, I think, is that the honeybees are not surviving nearly as well with 80% ethanol. And this may have been a trial run for their stable isotopes analysis, where they actually had to tether the flies and I think kind of more, not force feed, but give them the ethanol more directly that was radio labeled, or not radio labeled, just stable isotope labeled. I think that's the reason that the lifespan was slightly different. Again, not really the main take-home point. The main take-home point is that honeybees can't hang with the sauce. They can't hang with 80% ethanol like the oriental hornet can. Tethering is okay. It's not the best, but it's okay in both cases. It doesn't matter that much because their data from the stable isotope analyses happen across minutes, not days. So that's an important point. All right, so this next graph is going to show you how hornets are metabolizing the ethanol and not the honeybees. And this is where they're going to use the stable isotope. And we're going to look at two different graphs, one just for the oriental hornet and the other one for the honeybee. And these are two parts of a 6-panel graph, so it's even a decompression of one graph. But I want to explain to you very carefully what the data show. There's going to be two lines. One is for the stable isotope carbon labeled ethanol, and one is for the unlabeled ethanol. So it's gold and purple, those two lines. The axes are time in minutes and the stable isotope, delta 13C, in the exhaled CO2. Remember, we're measuring the carbon 13 in the CO2 that's in the airspace around these hornets that are tethered and consuming labeled ethanol. And so they're tethered, or they're in a confined space where they can collect the air. I'm showing a syringe there where they're going to pull out the air and then inject it into a isotope analyzer. For the hornets, we see this dramatic result that within minutes, literally within 10 minutes, the hornets have metabolized this labeled ethanol and are producing radio-labeled carbon dioxide. The unlabeled ethanol, of course, is not producing any of this delta 13C carbon, whereas the labeled ethanol is. And they did the control experiments to show that it wasn't just evaporation or anything like that. It was actually exhaled CO2 from the flies. And again, this is in minutes. It goes out to 20 minutes here, this chart. And now if we look at the honeybees, we see a dramatic difference. They're not using the radio-labeled ethanol at all. And the point is that therefore the flies, their assumption is that therefore flies are not using or metabolizing ethanol in the same way that hornets are. And that this leads to the question, what is happening here? Now we have an interesting point where maybe all that Sophia and Iran set out to do, maybe all they set out to do is this study of dose response in the oriental hornet, and showing with stable isotopes that the oriental hornets are actually very alcohol tolerant and they're using it. And they thought, wow, we're going to publish this paper, send it off to PNAS or Science, Nature, whatever. And the editors were like, wow, that's cool, but what about the mechanism? You could easily go in to do some comparative genomics here. And so they basically got sent back to the drawing board. But fortunately, they were able to cobble together the story from published information. And so they didn't actually have to do more sequencing themselves, but rather mine some databases to get at the comparative genomics, which I think did enhance the paper quite a bit. And it made the paper even more evolutionary focused than the ecology story that they told earlier, I think. So I put the kind of disgruntled face that might have been, I'm sure they were probably pretty disappointed if they got the letter back from the editor that said, nice, but we'd like some more work. And I'm not honestly sure that this is quite exactly the way it happened. I may be transcribing this story from another paper, but this is a pretty common story in science publication. So, so Sophia and Naran went back to their computers and did some comparative genomics. And I just wanted to highlight a few sentences from the paper that indicate this. They mined the genomes of other members of the genus Vespa by searching for such gene duplication events. And they used NCBI. And they found that all paralogs are encoded on the same chromosome, suggesting that they originated from a tandem duplication event, which is another way of saying unequal crossing over. Once you have this tandem duplication, that can lead to even more duplication on the same chromosome. Also, in that, I think in the next paragraph, they said that they also confirmed this gene duplication was also present in their model species, Vespa orientalis, which did not have complete genome sequences. So they're like, what are we going to do? But fortunately, there was some transcriptome information available from publicly available sequence reads. And they were able to use that information to find that there were two transcripts of the alcohol dehydrogenase NADP plus gene in Vespa orientalis. So I think for my students, what's cool is this really feels like a COVID project for students. A couple of years ago, it's like just go home, work on something, and you can get to something very interesting and relevant just by studying the publicly available data from NCBI um, and other sources. I don't know exactly where they got the transcriptome information, but they have a great method section here, and they had some databases there. So this is a phylogeny or a gene tree of ADH that they were able to produce from this deluge of genome information that they were able to find on NCBI. I want to point out that this is a gene tree which mirrors the species tree, but it's complicated because the species may show up in different places because different genes will have slightly different groupings. These genes are paralogous, they're copies of the same gene in the same species, but the genes are evolving in a fashion called concerted evolution. That's a word they use in this paper. It's a little bit beyond explaining that right here, right now, what concerted evolution is. Suffice to say, though, that once you have a gene duplication, those copies will evolve in their same, along their own lineages, not on their own species, but along their own dynamics. And we see that Vespa velutina, for example, has a gene copy that's grouping with or is ancestral to a bunch of other copies, but then Vespa velutina also has one that's sister to Vespa mandarania up at the top. But then look at the bottom, Vespa mandarania is also right next to Vespa velutina. So gene trees are useful in phylogenetic reconstruction, but it can present some complexity, but that complexity is actually very interesting because it tells you about the evolution of gene families. This part gets fairly complicated. I know students out there, but this begins, some of their big reveal is that they were able to put from their transcriptome data Vespa orientalis right there into this gene tree and show that Vespa orientalis also had two copies of the alcohol dehydrogenase, even though they didn't have complete sequence data. I want to point out that it says contig 1 right next to Vespa orientalis and contig 2 right next to Vespa orientalis. I'm going to explain what contig means next. The rest of the genes here have pretty long call sequences, and those would be useful in NCBI to get to that exact sequence. Again, the red are ADH sequences from the oriental hornet, and the oriental hornet is shown below there. So back to some glossary notes here. A contig is a set of overlapping DNA segments that represent a DNA region. This word is used a lot in sequencing because often there's a bunch of small reads or short DNA sequences that are assembled into one larger contiguous stretch, and that's where the acronym contig comes from. I also want to point out parsimony. Remember, this is a class in evolution, so I have to present some pretty important vocabulary, like parsimony is the simplest answer. It's in phylogenetic reconstruction, it's the phylogenetic tree which requires the fewest number of character state transitions. And a gene tree, what we just saw, is a phylogeny of genes that may largely represent the species phylogeny too, but it's really intended to reconstruct the evolutionary history of genes, which is usually similar to species phylogeny, but paralogous genes may form modiphylogous group within the species tree and next to other paralogous groups. So it presents some complexity, but it's also interesting. All right, also, since this is a class in evolutionary biology, I got to do some really basic stuff, like present the fact that Darwin was the first to make something like a phylogenetic tree in his 1838 Notebook B, where he sketched out what I consider the first phylogenetic tree. And above it, he says, "I think," which is pretty prescient. I think that little statement that he wrote in his notebook is a good reason to notebook. We still see this image. It's amazing. Also pointing out that there was only one figure on the origin of species, and it was this phylogeny, which is not often reproduced, although it is in our textbook. It's a little confusing, but it's still fairly prescient. The only confusing part is it seems like there's multiple origins of life, and even Darwin in the last paragraph suggests that maybe it was breathed into one or many, as his phrase. But I'm not going to go there because I think the genetic code pretty much settles that towards one origin of the life that we know so far on Earth. Our textbook has Darwin's figure, and it also has this other phylogeny, which is pretty good because it shows some lateral, some reticulation, some joining of branches. It also shows mass extinction events, some more complexity in the phylogeny. It has some data points there on the bottom, but I just wanted to point out that I actually got a tattoo of Darwin's phylogeny on top, combined with our textbook phylogeny. And if you don't believe me, there I am with my orange hat, so that you know that that's actually me and my tattoo of Darwin's phylogeny and the one from our textbook. So anyway, just thought that'd be fun tie-in. I also want you to remember that phylogenies can be depicted many different ways for graphical convenience. Sometimes you have less space or more space, or you want the words written to the left or the right. These are all the exact same phylogeny. And as an evolutionary biologist, you should become familiar and comfortable reading phylogenies in different forms, different patterns, I guess you could say. I'll just remind you too that the shared derived traits identify these monophyletic groups, and these are sets of taxa that include an ancestor and all of its descendants. And that's the concept of phylogenetic reconstruction. And you know that starts with synapomorphies, shared derived characteristics. Now I'm just going to go straight into the paper in this complicated figure. I know it's too complicated. I'm going to explain every little piece of it. And just stick with me here. But first, just kind of look at it. See what is going on. Ask yourself some questions. Why? Why? What is this? Where did it come from? How am I possibly going to understand this? I'm going to show you a piece by piece here. So this figure was produced by this program called Genesis Prime, or Genius Prime. I've never used it. This is a quote from their methods section. The sequence alignment was performed using blah, 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 implemented by Genius Prime, blah, 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 and this E-INS-I algorithm. So this is where students get lost with statements like this, but this is how they did it. Genius is a program produced by this company, and for those of you interested in going to into biotechnology, it's a data discovery, a graphical depiction program that's pretty useful, and they used it to produce this figure. I'm just going to parse this figure out. Take a deep breath because there's a lot to say here. First of all, remember that we're trying to find is alcohol dehydrogenase. Actually, maybe first of all, look at the 1 to the end is 35,895. That's the contig. That's the whole read. This is the region of DNA on that chromosome. And I forget which chromosome it is, but this is a contiguous read of sequence from a number of different wasps. We're going to start with Vespa crabo at the top, and that's the European hornet, which is different than the oriental hornet, which is the one that they did all the dose response curves and stable isotope ethanol experiments with. But European hornet's fairly similar to Vespa orientalis, and that's the first one. The contig is fairly continuous here. There's only a few gaps, which are the thin, narrow parts of that line, as I'll show you later. Within this contig, on purpose, there are 4 copies of alcohol dehydrogenase. They're in blue. Each blue arrow is an exon of a single copy of alcohol dehydrogenase. So there's 4 copies of ADH. Each copy has, if I count them up, 6 exons. 1, 2, 3, 4, 5, 6. If you count the one at the end, maybe 7. But point is, they've got exons. The arrows, the arrow part, with the thin part of the arrow being an intron between exons. The arrow points in the direction of its read, that is at 5' to 3' or the opposite. In this case, ADH is all transcribed in the same direction, 5' to 3', and right next to, well, at the ends of this contig, really a signpost, like this is where you should sequence, are these 2 sequences. Random genes, E3 ubiquitin protein, ligase synovin, and the DNA replication licensing factor, MCM5. Those are just used to find the area of DNA and start sequencing. And then the assumption is that ADH is somewhere between those 2 genes. And there's other stuff there, some other coding sequences, which we don't need to go into. And other information where there's other nucleotides where it's a thick line. And then in a few places, you'll see that it's a thin line. And when it's a thin line, that's a gap. And the gap is something they just had to introduce to align the sequences across this whole set of hornets. This is our whole picture again. Still too complicated. I'm going to break it down, go all the way down to Apis mellifera, the honeybee, and then build back to the whole picture here. So the honeybee, Apis mellifera, first thing you should notice is it has only 1 copy of alcohol dehydrogenase. That's it. It still has the 2 signposts. This is where you should sequence. It's got some other coding sequence in there. It's got a huge gap and some other smaller gaps and some other nucleotides, those thicker parts of the contig. But the main part is to take home is the honeybee has only 1 copy of this alcohol dehydrogenase compared to the hornets, which had 4. Now we go back to this, and one of the things that they found in their data sleuthing is that Vespa velutina had another copy of ADH that was sort of undescribed until they started doing this genomic mining. They're like, wait, there's another one in there. Nobody said, so that becomes a part of their result. Not a huge result for our purposes right now, but just highlights the effect of gene duplication and maybe how copy number itself can point towards some evolutionary relatedness. I'd like to point out this huge gap that was necessary to achieve this sequence alignment, and the more gaps, the more distantly related from Vespa crabo other organisms are. That's one piece of information you could use from the gaps that are necessary to do this. Remember, the Genius program is the one that produces this sequence alignment with these visualization tools, which are pretty cool. Otherwise, you just have a whole bunch of ACs, Ts, and Gs, or amino acids, and what do you do with that? It's much easier to look at exons and introns and gene sequences and say that this is one gene or another gene. All right, so some more glossary notes. Synteny is a shared order of sequences or genes between orthologous or paralogous genes or whole chromosomes in related species, used to identify similarities and differences between species and to reconstruct ancestral genomes. So some people might describe that previous graph as a synteny plot or a sequence and alignment plot. But synteny itself could lead to islands of recognition between chromosomes that may lead to misalignment and could lead to gene duplication. A gap is a graphical and a computational necessity when aligning orthologous and paralogous gene sequences or chromosomes. And carbon-13 is a stable isotope of carbon with 6 protons and 7 neutrons, not the normal 6, which makes up about 1.1% of all natural carbon on Earth. Now I'm going to go back to our news article, since I've really kind of explained the results of the Wasp paper. I want to get back to the human story a little bit. Remember this, that ethanol is degraded by alcohol dehydrogenase and produces acetaldehyde. In humans, ADH2, as also known as ADH1B, I know, it's a pain, but that's just the way it is. It plays a key role in metabolizing ethanol in the liver. There are 3 different forms of ADH1B, so it gets more complicated. Star 1, ADH1B star 1, is the slowest at metabolizing ethanol among these forms and is the most prevalent globally, except in many parts of East Asia. I'll just let that sink in. Humans, their ADH2 is known as ADH1B, but ADH1B has 3 different isoforms. ADH1B star 1, the first isoform, is the slowest at metabolizing ethanol, and it's the most prevalent globally, except in many parts of East Asia. The gene ontology or nomenclature is difficult here. I could have just said ADH1B star 1 is the slowest at metabolizing ethanol, and then said, but there's other forms, ADH1B star 2, which is faster. So we'll see that next. In East Asia, the most common isoform is ADH1B star 2, which metabolizes ethanol much faster than ADH1B star 1. So that has consequences, and perhaps beneficial consequences for these populations. As a result of this fast breakdown, acetaldehyde dehydrogenase becomes saturated, and therefore acetaldehyde accumulates in people that have this form of the enzyme, ADH1B star 2. Faster acting enzyme accumulates more acetaldehyde. As a result, remember, acetaldehyde is toxic and leads to some negative consequences. Acetaldehyde produces this alcohol flush reaction. This is from Wikipedia. If you search on alcohol flush, you get an image of this person, and this is from informed consent. The person has drunk some alcohol and has this alcohol flush reaction. You can see the redness in their face. So as a result, ADH1B star 2 carriers might, I put in quotation marks, might be less inclined to consume large amounts of alcohol because it doesn't make them feel good. It makes them nauseous, just like everybody, but it happens faster for them because they're actually metabolizing the alcohol faster and producing more acetaldehyde quicker. I should say that, you know, the flush face is common with people that are drinking or alcoholics or people with alcohol use disorder. Um, After you consume a lot of alcohol, of course, you're going to accumulate more acetaldehyde, regardless of what version of ADH you have. But this ADH1B star 2 is very fast acting, and you get this flush response very quickly um, as a result. So ADH1B star 2 is by far the most consistent hit in genome-wide association studies for AUD, which is alcohol use disorder in humans. And the way to conceptualize that is that if you have it, you are less likely to drink or less likely to have AUD. That's still a hit in GWAS land and speak because the negative is just as important as the positive for trying to understand what's the genomic association with AUD. Here's one that is not associated, which is also interesting. Another way you could look at this is that ADH1B star 1 is probably more associated with AUD. This paper, or Rainier's paper here, isn't fully on that. I'm sure there's a whole nother um, um, publication canon about ADH1B star 1 and its association with AUD. Remember, though, we're studying this Wasp paper, and we're kind of just using Rainier's helping us expand this to the human condition, too. Um, Another important point that Rainier pointed out in his commentary is that genetic factors contribute to AUD risk with a heritability of 50%. 50%, I put it in red there at the bottom. I should also say there's not a single gene for alcoholism. Don't use that terminology, the gene for. It's always a genetic architecture, and ADHs are going to play a role, but certainly aldehyde dehydrogenases are probably going to play a role too in AUD. Social and psychological factors are just as important too. It's not a simple story, and that's why there's still very active research, part of which Dr. Spanagel is doing at Heidelberg University. I just wanted to say at the end of this presentation that AUD, or alcohol use disorder, is a condition that affects so many people worldwide, and it even affected me at parts of my life. And I just wanted to reach out and say that there is hope for all of you that are suffering from AUD, and things do change, and it's so nice to be freed from those shackles. But as we say in Alcoholics Anonymous, once an alcoholic, we always introduce ourselves as alcoholics, even after recovery, and recovery is never complete because it's a lifelong process. And I just like to say I'm free from the shackles of AUD now, but want to acknowledge that some of you are probably still struggling with this, and it's partly a genetic factor. It's not all a social factor and psychological factor. It's not your fault, but I feel like with the strong heritability of AUD, it's important for everybody to know that it's a risk to fall into, and but there are treatments, especially enzymes like anabuse, that can help you modulate alcohol consumption, and that helps with certainly therapy, and Alcoholics Anonymous help too. So reach out if you have any need to contact about that. Back to my take-home points. The title again is Tolerant and Efficient Metabolism or Metabolization of Extremely High Ethanol Concentrations by a Social Wasp. The highly efficient ethanol metabolism in hornets or oriental hornets is most likely due to a duplication of the alcohol dehydrogenase gene. I know we saw 4 copies, but at least they know what they found in their transcriptome analysis, the data that they found on the internet. They found 2 copies. There may be more, but they at least know that they have 2 copies. Wasps may benefit from the 2 times higher energy content of ethanol compared to glucose, as well as from its antimicrobial properties when feeding on carrion. Yeasts benefit through dispersal to new sources of glucose. Wasps especially actually use the energy from ethanol, and they showed that with their stable isotope analyses. AUD in humans may be partly explained by slow ADH enzymes, partly, but there's more to that story, I'm sure. While ADH copy number and allelic variation may explain hornet ethanol tolerance, humans may have many more copies of ADH and cannot tolerate anywhere near 80% ethanol. So there must be other factors at play. Thanks so much for the authors of the study, Sophia and Iran, and other authors that were there. Sorry to ignore the people in the middle of the author sequence, but this is a fascinating paper that I think is of great interest to my students and certainly to me. And thank you, Rainier, for producing such a good commentary that really helped me place this target paper in the context of the human condition, and really appreciate all of that effort. Okay, thank you.